So I want to welcome everybody to today's SIP Spring webinar. SIP is the Society for Immune Polymorphism. For those of you who are not familiar with the Society, we're an international membership organization dedicated to understanding genetic and functional variation of the vertebrate immune system and the immune system's role in evolutionary biology, disease, and health. If you're not yet a member of the Society, you can join by going to tinyearl.com to join the SIP. Today's webinar is titled HLA Region Assembly and Genomics. If you have any questions for the speakers, please enter them in the Q&A. And I'm very pleased to introduce our moderator for this webinar, Dr. Janelle Noble. And I'm going to turn it over to Janelle.
Thanks, Steve. Well, it is my distinct pleasure today to welcome Dr. Marco Colonna to give our April SIP webinar. Marco is a professor of pathology and immunology at Washington's University in St. Louis. He's originally from Italy, and his career path took him through Roswell Park and Dana-Farber and the Basel Institute and landed him, fortunately for us, in the United States here in St. Louis. He has a very long and distinguished history, which I won't go into because I want to give him time to present the very exciting data. But the most recent award that, well, I think it was probably the most recent award that you won, Marco, is the Rose Payne Award from ASHI, which is a very big honor and very well deserved. And without further ado, I'm going to just turn it over to you, Marco. Welcome.
All right. Well, thank you very much. Let me see if I can share my presentation. First of all, I hope you can see it. Okay. Okay. And can you see my presentation well?
Yes,
Right.
I can.
So first of all, I'd like to thank you very much for this invitation to participate to this webinar. Steve and Janelle, thank you very much. I'm really delighted to be here. And thank you also for the opportunity to share some of my scientific trajectory that took me from alloreactivity to neurodegeneration. So these are my disclosures. And I'd like to start by saying that my scientific career started in 1987. And at that time, Pam Bjorkman, Jack Strominger, and Don Wiley made a major breakthrough in immunology by defining the crystal structure of MHC class I. Now, this was the first direct evidence that MHC class I was present in a peptide, and they provide evidence that the peptide was inside cleft created by alpha-1 and alpha-2 domains. And of course, this led to further discoveries, in particular, this solving or crystal structure of a T-cell receptor together with the MHC, which highlighted how the T-cell receptor can recognize peptides within the MHC. So, of course, these discoveries have many ramifications, but one important ramification was the understanding of the molecular basis of alloreactivity. It became clear that T cells, through T-cell receptor, not only recognize foreign peptides presented in the context of MHC class I, MHC class II, but can also recognize peptide in the context of allogeneic class I or class II, which will lead to alloreactivity. So in 1990, I joined the Strominger lab, and my initial idea, my initial goal, was to try to contribute to understand the mechanism by which the peptides are loaded onto MHC class I. But then I became interested in a different project that was quite unique. A group of Italian scientists led by Lorenzo Moreta had found that NK cells are capable of alloreactivity, or recognizing allogenic targets. And this observation was really enigmatic because NK cells were not known to express receptor for MHC. And so it was unclear what was the basis of this alloreactivity. And some genetic studies have suggested that perhaps the target recognized by NK cells was within the MHC. And so my goal was to try to understand which genes within the MHC was recognized by NK cells. And so I looked for polymorphism in MHC class 1, MHC class 2, and also in the complement region of MHC to try to identify polymorphism that would segregate with NK cell alloreactivity. And after 2 or 3 years of very hard work, I realized that individuals that were susceptible to killing by one NK clone all express CW1, CW3, or CW7 alleles. When I look at the sequence of these 3 alleles, I realized that they all share an epitope in the alpha-1 domain, a position 7780, characterized by the presence of serine and asparagine. And I will call this epitope serine 77. I also noticed that within the HLA-C alleles, this region, 7780, was dimorphic. HLA alleles had either serine 77 or asparagine 77. So I look at the individuals that are susceptible to killing by NK1, by this clone, and there were all CW1, CW3, or CW7. And I noticed that all these individuals express serine 77, either in a homozygous or heterozygous configuration, but they did not express the alternative epitope, asparagine 77. And so I came up with this hypothesis. I hypothesized that perhaps these NK clones express an inhibitor receptor for HLA-C alleles sharing asparagine 77. And so when the target expresses asparagine 77, the inhibitor receptor was engaged, and therefore NK cell would not kill. But when the asparagine 77 was missing and HLA-C alleles were expressing the alternative allele, this was unable to engage the inhibitor receptor, and therefore NK cell would kill the target. So in other words, alloreactive NK cells were recognizing the absence of a certain epitope of HLA-C. I look at the second NK clone, and I noticed that the second NK clone, individuals susceptible to a second NK clone, were all CW4, CW5, CW6. These individuals expressed the asparagine 77, but lacked the serine 77, the alternative epitope. And so again, I hypothesized that the second clone expressed an inhibitor receptor for HLA-C alleles sharing serine 77. And in the absence of this allele, NK cell will kill the target. So this led to our first important paper that showed that NK cell reactivity can be elicited across a polymorphism of HLA-C. Now I found a third clone of NK cells that was expressing an inhibitor receptor for a group of HLA-B alleles that share the presence of another amino acid, isoleucine 80, in the alpha-1 domain, approximately in the same region of HLA-C. And so in the absence of these HLA-B alleles, cells were killed by this NK clone. And finally, I found a fourth NK clone that was also inhibited by the presence of certain HLA alleles. And so, of course, this led to the identification of different specificity, HLA-A, HLA-B, HLA-C, recognized by NK cells. But the major question was, what are the inhibitor receptors that recognize these specificities? To identify this receptor, I moved to the Basel Institute for Immunology in Switzerland. This institute was famous because of the work of Susumu Tonegawa, who discovered immunoglobulin gene rearrangement, and also for the work of George Kohler, who together with Cesar Milstein discovered monoclonal antibodies. And so at that time, we only knew 2 major receptors expressed in NK cells. One was the Fc receptor, specific for immunoglobulin, and one was a receptor called 2b4. Both of these receptors were members of the immunoglobulin superfamily. And so I hypothesized that perhaps NK cells express additional receptor immunoglobulin superfamilies, some of which could be receptor for MHC, for HLA, A, B, C. And so after about a year of work, I identified 4 different receptors, which are now called killer cell Ig-like receptors. They're specifically expressed in NK cells. And these receptors were actually the inhibitor receptors specific for HLA-C, HLA-B, and HLA-A epitopes that I had previously identified. Now, we also found that these inhibitor killers acted by recruiting in their cytoplasmic domain protein tyrosine phosphatase SHIP1 and SHIP2. And this phosphatase initiate a cascade of dephosphorylation that inhibits cell activation. Now, we and others also identified a second set of receptors, which are quite unusual because these receptors were activated. And they activated by recruiting this adapter called DAP12 that was discovered by Louis Lanier. And this adapter was recruiting the protein tyrosine kinase SYK. So NK cells were controlled by a balance of inhibitor receptors and activating receptors, which mediate dephosphorylation and phosphorylation of downstream targets. Now, all these receptors were encoded by a cluster of genes on human chromosome 19, which we now call the leukocyte receptor complex. And by homology search, I found a second group of receptors called the leukocyte Ig-like receptor, or LILRs. Now, these receptors were quite similar to the killers, but they were expressed mostly on myeloid cells and not on NK cells. And the 2 of these receptors, specifically LILRB1 and LILRB2, were also recognized in MHC class 1. This receptor, however, was different from the killers, at least in 2 different aspects. First of all, they didn't recognize specific epitopes of HLA, but they recognized all the HLA molecules, HLA-A, B, C, E, and G. And later work by Pam Bjorkman, who co-crystallized this receptor together with MHC, demonstrated that this receptor recognized the beta-2 microglobulin and the alpha-3 domain of class 1 molecules, which of course are much more conserved. And the second important aspect that we notice is that these receptors, LILRB1 and LILRB2, are also expressed on myelomonocytic cells, including macrophages, dendritic cells, and monocytes. So this led to the idea that the recognition of missing cell, the recognition of missing MHC, is not a unique feature of NK cell, but it's also a feature of myeloid cells. And finally, also by homology search, I identified additional activating receptor expressed on myeloid cells. This receptor also recruited DAP12 and SYK and activated myeloid cells. And so I call this receptor triggering receptor expressed on myeloid cells. Now, after identifying all these receptors, we obviously became interested in understanding the impact of KIRs, LILRs, TREMs in immune responses. And for some time, we investigated their function in a number of different situations, like cancer or autoreactivity, and so on. But then, in 2013, our focus changed quite dramatically based on genetics studies, in particular on the genetics of Alzheimer's disease. Genetics has studied sporadic Alzheimer's disease, which is a major form of Alzheimer's disease for many years, since 1992. In 1992, the first DNA polymorphism associated with risk with Alzheimer's disease was identified in the apolipoprotein E4 alleles. But since then, a number of different risk factors have been identified. And we noticed that the vast majority of these risk factors were not expressed in neurons, were not expressed in oligodendrocytes, were not expressed in astrocytes, but were actually expressed in microglia. suggesting that microglia has an important function in controlling the progression of Alzheimer's disease. And we were particularly excited when we knew that when we found in 2013 that one of these DNA polymorphism associated with Alzheimer's disease was a polymorphism of the genes that we had cloned, TREM2. In fact, a hypomorphic variant of TREM2, the R47H variant, was found to increase the risk of Alzheimer's disease from 3 to 5-fold. As you know, Alzheimer's disease is the most common form of dementia, characterized by memory loss and declining cognitive function, and age is a major risk factor. And because of the age of the population, of course, this is becoming a major problem, major medical problem. And in fact, millions of people over 65 years old are affected by this disease. Now, according to the amyloid hypothesis, the first event in Alzheimer's disease is the abnormal proteolysis of a protein expressed on neurons called amyloid precursor protein. When this protein is cleaved by base-1 and by gamma secretase, proteolytic enzyme, they lead to the generation of a peptide called A-beta peptide, which is prone to aggregation and form aggregates that tend to propagate. So the first event in Alzheimer's disease is the generation of these amyloid plaques in the extracellular milieu of the brain. Accumulation of these plaques induces neuronal stress, and stressed neuron will then phosphorylate a microtubule associated protein called tau. Tau will precipitate and form these neurofibrillary tangles that eventually will kill neurons and cause cognitive impairment. We know that microglia are the major brain macrophages and the major immune defense in the brain in a steady state. They have this typical ramified morphology. And when we look at brain section of post-mortem specimen of patients with Alzheimer's disease, we see that amyloid plaques are entirely surrounded by microglia, which is trying to encapsulate amyloid plaques and prevent their spreading. We also demonstrated that TREM2 that we identified in myeloid cells in a peripheral immune system was actually highly expressed in microglia and was specifically binding to phospholipids. So we asked a question, whether TREM2 impact microglia response to amyloid plaques. Now, mice don't develop Alzheimer's disease, so we used a mouse model of Alzheimer's disease. It's a transgenic mouse model that expresses human APP with 5 different mutations that facilitate the generation of amyloid plaques. And so when we look at these mice, we see that these mice develop plaques in the brain. You can see them in green. And these plaques are entirely surrounded by microglia, who's trying to encapsulate the plaques. But when we look at mice that lack TREM2, we see that microglia is unable to form these enclosures of plaques. Now, when we look at the morphology of plaques, we see that plaques usually are pretty dense, but in the absence of microglia, they tend to spread out. They become more fibrillar, and they're more damaging. In fact, we can look at the damage in neurons, and particularly we can look at axonal swelling. Here you can see axonal swelling stained with N-terminus APP. You can see that around the plaques in red, you have some axonal swelling, but in a TREM2 deficient mice, where you have no microglia around the plaques, you have much more axonal swelling. We also developed mice that express the human variant of TREM2, which is associated with Alzheimer's disease. And we found that in this mice, microglia has a similar defect that we've seen in TREM2 deficient microglia. We're also interested to understanding how TREM2 can sustain microglia activation and response to the beta plaque mechanistically. As I mentioned, TREM2, like all the innate immune activating receptor, recruit this adapter protein, DAP12, and DAP12 then recruit the protein tyrosine kinase 6. And we know the SIK activate a signaling pathway, which involves PI3 kinase, AKT, and ultimately result in activation of the mTOR pathway. Now, mTOR is particularly important in promoting cell growth, in promoting protein synthesis, and also in providing energy to the cells, all features that are required for a response. And indeed, we found that in microglia lacking TREM2, DAP12, or SIK, we see a defect of phosphorylation of AKT, and we see also a defect of phosphorylation of NDRG1 and S6 kinases, which are targets of mTOR. So based on these results, we hypothesized that during Alzheimer's disease, and in general during neurodegeneration, a number of molecular patterns are released. First of all, extracellular aggregate of amyloid, but also apoptotic cells, apoptotic neurons, ATP released by dead cells, damaged DNA, damaged myelin. And all these molecules activate scavenger receptor and phagocytic receptors on microglia, then induce microglia activation, and facilitate encapsulation and phagocytosis of amyloid plaques. However, this process requires protein synthesis, requires energy. And this energy, this protein synthesis, is provided by the TREM2, DAP12, SIK receptor complex through activation of mTOR. In the absence of this pathway, microglia is unfit and therefore is unable to sustain activation for a long period of time. And therefore, individuals that develop plaques in the absence of mTOR or with the effect of the function of mTOR will eventually progress more rapidly towards Alzheimer's disease. So based on this data, we hypothesized that perhaps we can use agonistic antibody to activate TREM2, and this can be beneficial in AD pathology. So we collaborated with a number of companies that have developed monoclonal antibodies. And in the interest of time, I'm just going to show you one set of experiment. Here we generated transgenic mice that express the common variant of TREM2 or the R47H variant of TREM2 that is associated with Alzheimer's disease. And we crossed these mice, transgenic mice, with a 5xFAD model of Alzheimer's disease accumulates plaques. We waited until these mice developed plaque at 5 months of age, and then we started injecting them with an agonistic antibody anti-TREM2 that activates microglia. We did this injection for every week for 3 months, and then at the end of this period, we look at the behavior of the mice and we look at the brain pathology. Now, what you can see here in this brain section, you see in blue, you see the plaques. And as I mentioned before, around the plaques, you see this axonal swelling that you can see here in red. And in mice treated with control antibody, you see a considerable amount of axonal swelling around the plaques. But when you look at mice that have been treated with anti-TREM2 antibody, you see a reduction of axonal swelling that is statistically significant. So we also showed that treatment with the antibody can reduce abnormal behavioral traits in these mice. So a variant of this antibody has completed phase 1 clinical trial and also a phase 2 clinical trial in patients with Alzheimer's disease. Now, unfortunately, the clinical trial didn't meet the primary endpoint, which was a reduction, the slowdown of cognitive impairment. But we're learning a lot from this clinical trial. For example, we learned that in there's low antibody penetration in the central nervous system when we infuse this antibody IV. And so there's a number of attempts now to improve antibodies and also to generate most molecule to have an effective microglia activation in Alzheimer's disease. But going back to our background on NK cells and T cells, what I show you is that activation of NK cells, and we know also from T cell biologists that depends not only on activating receptor, but also on inhibitor receptor. And so this is also the case for microglia. In fact, what I show you is that microglia can be activated through TREM2, but we also know that microglia express a number of inhibitor receptors. And so the question we ask is then, as an alternative approach, can we activate microglia by blocking inhibitor receptor as we do in cancer immunotherapy for T cells? And so, of course, because of our background, we concentrated on the LILAR inhibitor receptor, and we look at this receptor in microglia. We were also encouraged to look at this receptor because when we look at the latest genetic data on Alzheimer's disease, we found the polymorphism in this region, specifically in LILAR region, are also associated with risk for Alzheimer's disease, although with less penetrance than TREM2 or ApoE. So we identified 5 inhibitor receptors of a LILAR family, LILARB1 through LILARB5. They all, a member of the immunoglobulin superfamily, they all recruit protein tyrosine phosphatase SHIP1 and SHIP2. And as I mentioned, 2 of these receptors, LILARB1 and LILARB2, are specific for MHC class I. So we wanted to see which of these receptors are expressed in microglia. So we performed single nuclear RNA-seq from post-mortem specimens of controls and patients with Alzheimer's disease. This is a UMAP that shows all the nuclei that we have sequenced. And what I show you here is the cluster of nuclei that represent microglia. So we look at the expression of LILARB1 in microglia. LILARB1 is specific for MHC class I molecule. You see that it is highly expressed in microglia and is also highly expressed during Alzheimer's disease. We also look at the second receptor called LILARB4, which is not specific for MHC class I, but it's actually specific for ApoE. And I'll come back to this point in a few minutes. We look at the expression again of LILARB4 in the brain of patients. We find that it's selectively expressed in microglia, and most importantly, is strongly upregulated during Alzheimer's disease. In fact, when we look at brain sections of patients with Alzheimer's disease, this is the amyloid plaque, which you see in blue. In green, you can see microglia encapsulating the plaque. And then in red, you can see the microglia has high level of expression of LILARB4. And you can see it also as well here. So we ask the question whether LILARB4 can impact microglia response to amyloid plaques. So to address this question, we generated a new mouse model that expresses one of the 2 clusters of LILARs, of human LILARs. So this is a back transgenic model that expressed the telomeric cluster, which includes LILARB4. We took this transgenic mouse, crossed them again to a model of amyloid pathology, and performed a similar experiment that we performed before with TREM2. Now, this is just to show you that in our transgenic mice develop plaques. You can see here in blue. And you see the plaques are surrounded by microglia. And now microglia in these transgenic mice express human LILARB4. So we recapitulated exactly what happens in human. And now we take these mice, we let them develop plaques until they're 4 or 5 months old, and then we inject them with weekly injection of an antibody that recognize LILR-B4. And then after 8 weeks, we look at the pathology, we look at the behavior. Now, this just to show you that the antibody reaches very high concentration in the serum, also reaches some concentration in the brain, although the levels are detectable, but much lower than what you see in the serum. So again, there's a defect in the penetration of the antibody into the brain. And after the treatment, we take a brain section and we stain them with methoxyl-4, which detects fibrillar A-beta, or with 6010 antibody that detects also A-beta in a number of different forms. And you can see that in both cases, with both staining, we see a significant reduction of the load of A-beta plaques in mice treated with the antibody anti-LILR-B4. And this is true throughout different brain regions, in the cortex, in the hippocampus, and also in the amygdala here. And in data that I'm not going to show you in the interest of time, we've shown that this treatment actually can enhance the phagocytic ability of microglia against amyloid A-beta plaques, in particular amyloid fibrils. So we ask the question, why LILR-B4 can inhibit microglia in Alzheimer's disease? What is the mechanism? As I mentioned, work by Alex Zhang in UT Southwestern has shown that LILR-B4 is not a receptor for MHC class 1, but it's a receptor for ApoE, apolipoprotein E. And we know for several studies that amyloid plaques do not contain only A-beta fibrils, but they also contain ApoE that precipitates within the plaque. And so we came up with this hypothesis. We hypothesized that when the plaques are encapsulated by microglia, microglia expresses very high level of LILR-B4, but the plaques also express ApoE. And therefore, ApoE will interact with LILR-B4 and induce an inhibitory signal. So to test this hypothesis, we generated a reporter assay. In this assay, we take LILR-B4, the extracellular domain, and we convert the extracellular domain into an activating domain by generating a chimera between LILR-B4 and CD3 zeta, such that when LILR-B4 active domain now is engaged by a ligand, this will induce a signal that will induce calcium. And in this reporter cell line, calcium will induce GFP. So we treated this reporter cell line with increasing concentration of ApoE, and you can see that indeed, when we increase the concentration of ApoE, the reporter is activated. So indeed, LILR-B4 is the receptor for ApoE. Most importantly, when we perform this assay together with the anti-LILR-B4 antibody, we completely block the activation of the reporter. So this indicates that the antibody that we have generated is a blocking antibody that blocks the interaction between ApoE and LILR-B4. And blockade of this interaction results in release of microglia from an inhibitory signal and activation of phagocytosis. So what I show you is that LILR-B4 is an inhibitory receptor expressed in microglia, and particularly highly expressed in microglia around the plaques. And the expression of this receptor correlates with the amount of amyloid pathology. And what I show you is that we can, with an antibody, a blocking antibody against LILR-B4, we can activate microglia by interfering of the binding of LILR-B4 with ApoE on A-beta plaques. And ultimately, this checkpoint blockade can facilitate the clearance of amyloid burden by increasing phagocytic activity of microglia. So what I show you is that we can get therapeutic activation of microglia in Alzheimer's disease, not only by engaging activating receptor, by also blocking inhibitory receptor, we have obtained evidence that this is the case for LILR-V4, but we're also exploring other inhibitory LILRs. So in a final close of my talk, I like to mention that, and this is really for future perspective, is that we have expanded our knowledge of activating inhibitor receptors to neurodegeneration, to Alzheimer's disease particularly. But there's also evidence that HLA and also receptor for HLA, both on T cells and innate immune cells, may be important in other neurodegenerative diseases. For example, Parkinson's disease is a disease caused by aggregation in substantia nigra of aggregated protein called synuclein. Now, extensive genetics has been also performed in this disease, and among the various risk factors being identified, one of the risk factors is HLA-DR-B5. And indeed, individuals that have this particular allele of class 2 are more prone to disease. Now, work by Alex Sitter and also by David Salzer have shown that if you isolate T cells from patients with Parkinson's disease, especially patients that express DR-beta-1501, a subtype of DR5, they have specificity for alpha-synuclein peptide. Alpha-synuclein is one of the proteins that aggregates in substantia nigra and causes the death of neurons. So these experiments suggest that additional receptors can be evaluated in neurodegeneration. And so the take-home message of my talk is that we're understanding the HLA variants and their receptors, both on T cells and innate immune receptors, is key for developing new therapies for neurodegeneration, Alzheimer's disease, Parkinson's disease, perhaps also amyotrophic lateral sclerosis, and other neurodegeneration. So this is a message of my talk today. I'd like to make some acknowledgement. Throughout my career, I've been very fortunate to work with two very talented scientists, Marina Cell and Susan Gilfillan. We worked together from the time of the Basel Institute for Immunology, and we continue to work today together at the Washington University School of Medicine in St. Louis. They've been instrumental in all the studies and all the results I presented. I also like to mention my MHC mentors. I started in a lab in Genova, was collaborating with a group in Buffalo, New York, led by Nobuo Tanigaki, G.B. Ferrari, and Roberto Tosi. Unfortunately, they're no longer with us. Jack Strominger was my mentor at Harvard. He's almost 100 years old, but he's still active and mentally very sharp. Emil Ulano has been my mentor at WashU. Unfortunately, he passed away. He was another giant of the MHC, particularly in antigen presentation. I have a group of people that work on neuroimmunology, and I also like to acknowledge some of former members of my lab who have contributed to this work. I have important colleagues at Washington University I'd like to mention. Wen Yokoyama discovered mouse NK cell receptor, and of course, we've been talking together for many years about the relevance of this receptor in innate immunity and beyond that. Yoni Kipperson and David Holtzman are very important collaborators for our work in neurodegeneration and neuroimmunology. And these are some of our industrial collaboration that, of course, are important to test our hypothesis in vivo in patients. So with that, I will close here, and I'll take any question. And thank you for your attention.
Okay, can people hear me? I'm hoping that people can hear me. Thank you, Marco, for a wonderful seminar. I learned a lot and took 3 pages of notes. And, I heard this talk back in October at ASHI, and I learned more this time after hearing it again. I think this is really fascinating stuff and incredibly well-timed given our aging population and how many neurodegenerative diseases are becoming more and more prevalent in the population.
Thank you.
So first question here is from Raj from UCSF. And Raj asked, what triggers changes in TREM2 activity in older adults that lead to Alzheimer's disease?
So I show you that TREM2 is a receptor for phospholipids, and we're still debating which phospholipids are recognized. We know that phospholipids are exposed on dead cells. So apoptotic cells that expose phospholipid can be an important trigger for TREM2, and they also increase during aging. We also know that apolipoprotein, like ApoE, can also expose phospholipids. And these have been shown in vitro that they can trigger TREM2 signaling. And ApoE is the major lipoprotein expressed in the brain, is also precipitating in the plaques, and is particularly abundant in amyloid plaques. So the accumulation of ApoE may be another important trigger of TREM2 that increases over age. There could be additional phospholipids that are exposed in other situations, but these seem to be the two major sources of ligands for TREM2: apoptotic cells, dead cells, and apolipoprotein E.
Okay, and I just want to remind people that if you have questions, please put them in the Q&A section of your Zoom screen. The next question we see is from Martin Myers. He says, do you know if the LILRB1/2 binding to class 1 alpha-3 domain is specific? And if so, is this a consideration for alloreactivity in cell therapies? For example, missing self.
Yep, yeah, so very good question. So so the crystal structure of LILRB1, LILRB2 with MHC class 1 has been solved by Pen Bjorkman. And so you can see all the structural detail in this beautiful paper that they published. In general, LILRB1, LILRB2 bind to the beta-2 microglobulin and the alpha-3 domain. The beta-2 microglobulin is invariant. The alpha-3 domain has very little polymorphism in the population. So in general, I don't think that LILRB1, LILRB2 are a major concern in alloreactivity. Having said that, the alpha-3 domain can be polymorphic, and so perhaps some variants may also block the recognition by LILRB1, LILRB2. In general, I think that these receptors are very important in situations in which you have complete lack of MHC class 1. For example, in tumors that are trying to escape T-cell recognition, and when you try to escape T-cell recognition, they downregulate beta-2 microglobulin, they downregulate the entire MHC class 1 molecule. And so in this situation, LILRB1, LILRB2 become important in recognition of target. So more in recognition of missing self, but not in recognition on allo recognition.
And I actually had a related question to that because you said that LILRB1 and 2 recognize lots of different MHCs, A, B, C, E, and G. Are there molecules that are more strongly associated or recognized by LILRB1?
Yes.
For example, HLA-C, do you see that binding more strongly than HLA-B or?
So no, HLA-G actually is the major binder of LILRB1 and LILRB2. And for that reason, HLA-G, as you know, is highly expressed in fetal trophoblast in the placenta. And so for that reason, LILRB1 and LILRB2, especially LILRB1, has been extensively studied in the context of pregnancy and in the context of maternal fetal tolerance. So there's a number of different studies that have shown that NK cells in the placenta express LILRB1, macrophages express LILRB2, and the interaction with HLA-G expressed by fetal trophoblast seem to be important in maintaining maternal-fetal tolerance. But having said that, it binds with a good affinity also with other molecules, but certainly HLA-G perhaps has the highest affinity.
Thank you. We have another question here from Raj, who asks, is TREM2 polymorphic? Are any variants associated with an increased risk of Alzheimer's disease, and how common are the variants?
Yes, that's a very good question. So the first variant identified, so let me actually, I didn't want to complicate that, but before TREM2 was found to be associated with Alzheimer's disease, early studies by Leena Peltonen, a geneticist in Finland, found that complete defect of TREM2 expression in few families in Finland is associated with very early onset dementia, which is called Nasu-Hakola disease. Now, this mutation, completely inactivating mutation, they delete the gene, they completely block the expression, and in homozygous configuration, they cause early onset dementia. These people develop dementia when they're 35, and usually they die right after that, when they're in the 40s or 50s at best. So there was an early evidence that there's some polymorphism in the population that caused a complete blockade of TREM2. But there's not a lot of attention because this disease obviously was a rare disease, was a rare genetic disease. Then in 2013, the first variant of TREM2 was identified by ENCODE in Iceland by Kari Stephenson. And they found the R47H mutation is associated with increased risk. Since then, additional variants have been identified. Many different variants have been identified, but the frequency is very low. It's below 0.5% in the population. So, and all the hypomorphic variants are associated with increased risk of developing Alzheimer's disease. So when you test the binding of TREM2 for phospholipids, all these variants reduce binding, and all these variants are associated with Alzheimer's disease. Only one variant has been identified that increases the binding affinity, and this variant is actually associated with protection from Alzheimer's disease. So there's a very clear evidence that this gene is polymorphic, and the polymorphism that reduces binding affinity for the ligand and reduce function are all associated with increased risk to develop Alzheimer's disease.
Fascinating. Okay, I am not seeing any more questions in the Q&A panel, and I don't see anything in the chat here. So I think that means, Marco, that your seminar was incredibly clear. And again, I want to thank you for joining us for this webinar and letting everyone hear this who may not have heard it at ASHI, and letting people hear it who did hear it at ASHI. And I will turn it back over to Steve, I think, who has a few concluding remarks here.
Okay, but before you close that, I would like to thank you again. And I also like to, the attendees, that if they have any questions, please email me. I'll be happy to answer the question after this webinar at any time. And if anybody's interested, if there's a postdoc or student interested in these topics, please contact me, and we can discuss potential collaboration and potential projects in my lab. Thank you.
And Marco, we had one more question come up while you were advertising for a postdoc, and that is from Jed Barger, who asks, do you believe similar trials can show promise for individuals with higher risk isoforms of ApoE, such as ApoE4?
Yes, it's a very good question. So far, the clinical trial has been done with populations that have lower risk to try to maximize the potential therapeutic effect of TREM2. But I think that with the new therapeutics that have been developed, both in terms of small molecules and more penetrating antibodies, I think it would be important also to look at populations that have higher risk, for example, the ApoE4 population. Certainly.
Thank you. And now with that, I think we have addressed all of the questions, so I will turn it over to Steve for concluding remarks.
Thanks, Janelle, and then thanks, Marco, for this great talk. I really enjoyed your historical perspective, and it was very educational. I want to thank everyone else who attended the webinar. I hope you enjoyed it and got a lot out of it. A recording of the webinar will be made available on the Society for Immune Polymorphism's YouTube page if you want to watch it again, or if you want to get your friends and colleagues excited about this work. The Society will be holding its next webinar on September 30th. The topic will be the genes that encode the T-cell receptors and B-cell receptors, and this work will be presented by Dr. Melissa L. Smith and Corey Watson, both from the University of Louisville. So we look forward to seeing you all in September. Thanks very much for attending.
All right. Thank you all.
Bye, everyone.